count of 3, just yell out Atlantic.
Hi, my name is Sabrina Chang. I'm an 8th grade student at Atlantic Middle School, and this is our STEM fair project.
Hi, my name is Rachel Chan, and I'm a student from Atlantic Middle School, and this is our project. Our question is, does the text color of a short story affect a middle school student's short-term memory?
Our hypothesis was that if blue text color was used for a short story, the amount of correct answers would increase by 20%.
Independent variable is the text color, and our dependent variable is the middle school student's short-term memory.
Our materials included participants who used their Chromebooks to answer a Google form. Our procedure was to gather the students, have them sit down, set a 2-minute timer, let them read a short story, and once the timer went off, we set a 4-minute timer for which they could answer text-dependent questions.
Control black text has an average of 86.66% correct answers, and our experiment 1 red text got an average of 94.66 correct answers. And our experiment 2 blue text got an average of 83.33 correct answers.
These are some of our pictures, and this is how we arranged our seats. For every single time we did our experiment, we had 4 participants at each end of the table. And here you can see our participant. She's reading a short story and she's answering the questions. Our conclusion for our experiment was that the text varies, or the data varies too much. As you can see here, it varies from 74% correct answers to 90%.
Hi, I'm Tom Leung. I'm on the school committee, and it's really great to be here at Atlantic to see all of the wonderful projects that these 8th graders have worked so hard on. I'm just so impressed with the quality of the work and the way they explain themselves and answer questions about their process and their findings.
My name is Paige Toland. Our question was, does changing the fruit affect the amount of extractable DNA? And we hypothesized that if we do change the fruit, the amount of extractable DNA will differ. Our variables, the independent was the strawberries, and the dependent was the other fruits. Our research paper was about the guy who first discovered DNA, which was Frederick Meister, and it's also about deoxyribonucleic acid, which is also commonly known as DNA, and that it's the molecule that carries genetic information throughout humans' bodies and throughout fruits. Our procedure was that we would take the fruits and we would crush them into a pulp inside of a Ziploc bag, and then we would add 6 grams of salt and 11 grams of dish soap, and we would add 150 milliliters of water, and we would mix that until it was evenly distributed, and then we would pour that over a paper towel, which was over a test tube, and then we would pour rubbing alcohol down the sides and mix it with a skewer until the DNA came out in strings. Our results were that mangoes and strawberries both had 0.01 grams of DNA. Bananas had the most, and raspberries had none. And then for our conclusion, we chose to experiment with extractable DNA because people don't have enough knowledge about it, and also something we could have changed was the amount of the type of salt that we used, and just in general, we wish we had more time. And for our future application, I think if we had done this a different time, we probably would have used different materials, and we probably would have used a microscope. And also, to put this into use for other people, for farmers and people who want more protein in their food, that's who we'd probably recommend this for.
My name's Fayla Morris, and these are our materials. Our materials were paper towels, test tubes, alcohol, strawberries, bananas, mangoes, and skewers. This is our data. We had raw data trials 1 through 3. Then we had our average mass of DNA per trial, and then amount of extractable DNA, and then average mass of DNA per fruit type.
Hi, my name is Kaylin Saavedra. Our STEM fair topic is on artificial intelligence. The question that was being tested was, can informational recognition be detected by AI if data is changed? And then our hypothesis is, if AI is given incorrect data, then AI won't be able to detect that it's incorrect. Variables that we were tested on was independent was if we put incorrect data, and independent was AI recognizing that it's incorrect. The constants, which were the things that were kept the same, was the computer, highlighter, notebook, pencil, and OpenAI websites. Two things I learned about the research paper is AI started as a simple question: can machines think, by Alan Turner, and there's different types of AI, such as OpenAI and generative AI and our AI to create different things.
Throughout the experiment, we used the materials of one computer, one pen, notebook, highlighter, and pencil. And our procedure was to collect data sets of changing selected information in the data sets, making a copy of the incorrect data and keeping one of the correct data, opening ChatGPT, clicking on add files and more, uploading the Excel spreadsheets, inputting incorrect and correct data sets. Then we ask ChatGPT and we input if there's any difference between the two data sets. After letting ChatGPT analyze all the data sets, Excel and spreadsheets, it report back to us, identifying the difference between the original and what we inputted. And we recorded the answers on a notebook. Some data sets that we showed was the data was what we inputted, which shows a trial of 10 trials. Showing here of the right and wrongs. 91.40% of it of what we inputted into the ChatGPT was right, and 8% of the data we placed into the ChatGPT was wrong. And a separate outcome was a hallucination. A hallucination was a response produced by an artificial intelligence program or a tool that appears to be accurate or plausible that contains inaccurate or misleading information. Here's all the data sets here, and it shows, and here's some pictures of us inputting the data, ChatGPT giving back the answers, and unexpected outcomes. For the results was that data made data of its own. ChatGPT detected most differences between the data sets, as well as ChatGPT missed very little data.
We chose artificial intelligence because we wanted to see how reliable AI is. Also, AI is used worldwide. Estimated over 1 billion people using AI around the world monthly. And we wanted to see. Oh my God, what was I going to say? Oh my God. And our hypothesis was, if AI is given incorrect data, then AI won't be able to detect that it's incorrect. But that wasn't necessarily supported with 4 out of the 10 trials making data of its own, which is called a hallucination in AI, which is a response produced by an artificial intelligence program or tool that appears to be accurate or plausible, that contains inaccurate or misleading information, almost like AI has like a mind of its own. So the data wasn't so what it made was, it wasn't directly pulled from the internet. It made its own, which is not necessarily supposed to do. And then what surprised us the most was the unexpected data, and that's basically it. And to remember that artificial intelligence isn't something of its own. That the intelligence comes from us and things that are supported by us. To continue this project, I would use a different OpenAI website, and I would give it multiple math problems such as algebra, statistics, pre-calculus, calculus, and input it all at the same time to see how much it gets wrong and right.
But if I was continuing to talk about this topic and probing and exploring again, is that I would change the data set and put it into ChatGPT or any other AI-related websites, such as like putting grammar accuracies, essays, or just putting any like school-related stuff, and etc.
Our experiment is about DNA. So DNA is this invisible code which is inside all of us, and it really depends on the person. It varies from person to person. So our question of the experiment was, does the temperature of the alcohol affect the DNA yield? And the answer is really yes, it really does affect the temperature. Because when we did the experiment, we also saw the results. The results were really, really obvious because the cold temperature alcohol breaks open the cells and it helps us to extract DNA more efficient than the hot temperature.
For the variables, we did the alcohol temperature independent variable. For dependent variable, we did amount of visible DNA. And for the constants, we did volume of alcohol, amount of sample, salt, concentration, type of sample, extraction method. And this is a research paper, which is basically the procedure, but very longer. For the data, we did the temperature of experimental groups, which you can see that we did 5 trials, and this one is probably the trial free is probably the highest. And for DNA sample collection counts, we did 3 trials, which in the trial 1, which was the warmest, there was basically 0 white substance, the DNA amounts. But for this one, the trial 3, which was the lowest trial 3, it was like maybe 5 DNA samples. And for conclusion, our experiment was successful because we could extract DNA from strawberries using dishwasher salt, water, and rubbing alcohol. And we saw a white cloudy substance on the mixture that we made. So our future application would work for, our experiment would work for the people, scientists that work on animals or genetics, or the people that solve crimes.
The materials are really easy to do, and you can also buy them at any shop. So the simple materials are strawberries. Of course, you can use many different types of fruit, but strawberries intend to be the most easiest and efficient way to extract DNA. They also intend to have more DNA than other fruits. The other one is rubbing alcohol. Rubbing alcohol, of course, this also affects strawberries because rubbing alcohol can easily break open the strawberry cells and helps us extract the strawberry DNA. You can also use many different types of alcohol, but rubbing alcohol helps us do better than regular alcohol. The plastic Ziplocs, so we can for plastic Ziplocs is needed because we can carry around and do our DNA extraction. Salt, dishwashing soap, and water. So with these 3, we can combine them and make a solution out of them to put it in strawberries. The procedure is also easy. We just take some strawberries and put them inside a Ziploc bag. Then we mash it up for like 2 minutes, and then when it's totally mushed up, we take 2 teaspoons of dishwashing soap and 1.5 teaspoons of salt and 1,000 milliliters of water. Then we make a solution out of all of them, and then we pour it inside the mixture, and then we take and then we shake it up a little and and we wait it for 2 to 3 minutes. After that, we take the Ziploc which has the mashed strawberries in it, and then we take a coffee filter paper and we put it on top of a cup or a bottle. We then pour out the mixture, DNA extracted. This also impacts very future applications because, as Archie said, people who work in the industries and everyone who has works in the science forensic department can use this method to find criminals who have gotten away with their crimes. And also, this helps the scientists to study more about genetic information on living organisms like plants, humans, and animals. And that's it. Thank you.
Hello, I'm Spencer. This is Caden, and this is our project on Rubik's Cubes.
Okay, so our question is, can the ability to solve the Rubik's Cube improve recognizing colors and patterns? And then our hypothesis was that if you know how to solve a Rubik's Cube, it will improve all of this.
Our data shows that people who had solved the Rubik's Cube did on average better. The people who had not solved the Rubik's Cube had about on average a 6.9, and the people who had solved the Rubik's Cube had about a 9 out of 10 average. We decided that we had enough data because it would show that we had enough data to have a confirmation that we had enough people and we were set to have our representation.